Hello, my name is Sophia Fagan, and today is January 8th, 2020. Today, I will be talking to you about the reproductive system of the human body, more specifically, the determination of gender, genes versus hormones. The sex organs of both males and females consist of three sets of structures, which are the gonads, the internal genitalia, and the external genitalia. The gonads are the organs that produce the gametes, which are the eggs and sperm that will come together to form new individuals. The internal genitalia are comprised of accessory glands and the ducts that connect the gonads with the outside environment. Lastly, the external genitalia include all of the external reproductive structures. The photo on the right depicts the gametes of the egg and the sperm being the slender, thin structure on the left side, and the egg being the large sphere on the right. How we develop sexually is programmed in our genes. Each nucleated cell of the body, except for the eggs and sperm, has 46 chromosomes. This is called the diploid number of chromosomes because there are two pairs of chromosomes that it comprises. This includes the 22 matched pairs of autosomes plus the sex chromosomes. It is for this reason that we are considered diploid organisms, indicating duplicated chromosomes. The two sex chromosomes are either X or Y, and themselves contain the genes that coordinate the development of the internal and external sex organs. When the egg and sperm unite, this creates a unique set of 46 chromosomes with matching pairs of chromosomes, each set coming one from the mother and one from the father. You can see in this image 22 pairs of chromosomes in the purple box, and then the pair of sex chromosomes in the blue box. The sex chromosomes that an individual inherits will then determine the genetic sex that the person is. Genetically determined females have two X chromosomes and therefore are denoted XX. One X chromosome comes from the mother, and one X chromosome comes from the father. Genetically determined males have one X chromosome and one Y chromosome, and are therefore denoted as XY. The X chromosome comes from the mother, and the Y chromosome comes from the father to create a male offspring. It is this Y chromosome that provides the necessary information for development of the male reproductive organs. You can see here that the X-labeled egg from the mother and the X-labeled sperm from the father create an XX female offspring. You can also see that an X-labeled egg and a Y-labeled sperm create an XY male offspring. Early in development, it is very difficult to determine the sex of the embryo because the reproductive structures have not started to develop yet. Therefore, early in development, the embryonic tissues are called bipotential because they cannot be morphologically distinguished as being male or female. The bipotential internal genitalia has two pairs of accessory ducts called the Wolffian ducts and the Mullerian ducts. Once the embryo further develops, the sex of the embryo will determine which of these ducts will develop and which will degenerate over time. The sex determination of an embryo also has to do with the absence or presence of the sex-determining region of the Y chromosome, otherwise known as the SRY gene. If the SRY gene is present, then the bipotential gonads will go on to develop into testes, therefore becoming a male. If the SRY is not present, the gonads will develop into ovaries. This diagram nicely brings together what can come of the gender of the embryo after this bipotential stage at week 6, depending on a number of different factors. As you can see in the top depiction, there are some early structures that begin to form at this week 6. But keep in mind that they are not yet gender specific. Soon, they will develop to be certain structures based on what the actual sex of the embryo is. You can see the Mullerian and Wolffian ducts, as we discussed before, as well as the gonad bipotential that we also discussed, and the kidney. So if the embryo is female, there will be no anti-Mullerian hormone present. So the Wolffian duct will be the one that regresses, and the Mullerian duct will then become the fallopian tube, uterus, cervix, and the upper portion of the vagina. The gondal cortex will proceed to become the ovary, and the gondal medulla will be the one that regresses. If the embryo is a male, there will be an anti-Mullerian hormone present, causing the Mullerian duct to regress and the Wolffian duct to continue to form the epididymis, vas deferens, and seminal vesicle. You also see that testosterone is present for this portion. Testosterone is a male sex hormone, but it will not be secreted until after the gonads have differentiated into the testes. Also for a male, the gonadal medulla will form a testes, and the gonadal cortex will regress. As you can see here, this is the second half of the figure that we just discussed, and this time it's looking further at the development at a week 10 embryo, and then at the bottom of the page at birth. Keep in mind that the female is on the left and the male is on the right. Starting with the female, the gonadal cortex then becomes the ovary, and the females have an absence of this SRY protein. I will repeat this. The females have an absence of the SRY protein that we discussed previously, which will result in ovary formation. The development continues under female-specific genes. There is also an absence of the hormone testosterone, which will cause the Wolffian duct, seen to further develop in males, to completely degenerate in females. At birth, the anti-Mullerian hormone, denoted as AMH, is not present, so the Mullerian duct continues to become the fallopian tubes, uterus, and the upper part of the vagina. On the right side, at week 10, the SRY protein is present in the male embryo, and this directs the medulla of the bipotential gonad to then develop into the testes. However, there is the AMH hormone present, which causes the Mullerian duct to degenerate. The testosterone that is present in the males changes the present Wolffian duct into now a seminal vesicle, vas deferens, and epididymis. There is also another hormone present here, which is DHT, which stands for dihydrotestosterone, and it is a derivative of the testosterone that in this case will control the development of the prostate. In this lecture, we have learned about how gender can be determined both by genes and hormones, and their presence or absence during development.